My name is Trace Smith, and I am accompanied by Mia Cassabat and Kaylin Gerald. This presentation for Dr. Benton's Chemical Engineering 4220 class is on twin genetics, Josef Mengelé's experiments. The book that we chose to read is Neanderthal Man in Search of Lost Genomes, written by Svante Pääbo, who is a Nobel Prize winning Swedish biologist and geneticist. It depicts Svante Pääbo's scientific journey, beginning with obtaining ancient mummy DNA and ending with sequencing the nuclear genome of Neanderthals. It also describes the challenges of sequencing genomes of ancient organisms and the technological advances that allowed for the Neanderthal nuclear genome to be sequenced. The book also incorporates ethical problems and considerations of uncovering ancient DNA of Neanderthals. Lastly, it celebrates the scientific insights uncovered through the genetic material of Neanderthals and what humans can learn about our human origins. The portion of the book that piqued our interest was Chapter 7, A New Home. At the beginning of the chapter, Svante mentions how the Max Planck Society he became a part of had Nazi roots, once called the Kaiser Wilhelm Society, doing weapon research and racial science. It also mentions that Josef Mengele was an assistant at the institute while conducting experiments on twins at death camps. We wanted to explore the ethics behind his actions as well as the scientific research he was conducting. We were also interested in how the sequencing methodology used in the book are used in current twin research, specifically the polymerase chain reaction, or PCR, which was used to multiply Neanderthal DNA, and the single nucleotide polymorphism, or SNPs, which were used to distinguish the differences between Neanderthals and humans, as well as estimate the split time of the ancient humans and Neanderthals. Now, Kaylin will discuss Josef Mengele and his experimentations.
Josef Mengele was a German physician and SS officer who conducted medical experiments on prisoners at the Auschwitz concentration camp during World War II. As mentioned before, he first became a research assistant at Hereditary Institute, but later became known as the Angel of Death for his research on twin genetics. Josef Mengele conducted his research in an attempt to uncover the secret behind genetics and prove the Nazi ideology. He wanted to know how genetic diseases originated and what factors affected them, and he also researched ways to differentiate between identical and fraternal twins. Mengele also looked into epigenetics and wanted to discern between genetic traits and those developed by the environment of the child. He was looking for a way to prove that there were genetic weaknesses in the makeup of Jewish or Gypsy people. Mengele's experiments were wildly unethical and lacked proper control groups, sample sizes, and statistical analysis. His research was often based on faulty assumptions about genetics and human behavior corrupted by the Nazi views. The ethical principles of modern medical research emphasize the importance of informed consent, respect for human rights and dignity, and the pursuit of scientific knowledge that benefits humanity. Mengele's experiments violated all of these principles, and they serve as a stark reminder of the dangers of unchecked scientific research and the importance of ethical oversight in scientific inquiry. Now Mia will discuss current twin research.
Twin studies are still used today with numerous ethical principles put in place because they are essential for measuring the contribution of genetics to diseases, mutations, and more. This picture shows the differences during fertilization of identical or monozygotic twins and fraternal or dizygotic twins. Monozygotic twins originate from the same egg and are split in half after a single sperm fertilizes the egg and are therefore derived from a single zygote. Monozygotic twins share the same set of chromosomes and genes. However, there may still be differences that can be observed in some identical twins, including differences in susceptibility to diseases. This is particularly what Mengele was interested in studying. On the other hand, dizygotic twins arise when two egg cells are fertilized at the same time by two sperm cells. In this case, each fetus has its own placenta and grows independently. Because of this, they only share 50% of the same genes. However, in most modern-day twin studies, researchers look for identical twins that grew up in the same environment so that both factors can be used as controls. Recently, scientists have become interested in studying X inactivation in female twins. Because females are born with two inherited X chromosomes, one from mom and one from dad, X chromosome inactivation, or XCI, has to occur to transcriptionally silence one of the X's. This is a random process that occurs during embryogenesis and is maintained during cell replication. In most cells, the amount of active genes from the X chromosome is equally split between the paternal and maternal X chromosome inherited. However, scientists began to notice that the X chromosome inactivation becomes skewed towards one parental X as the female ages. This is an important field of study because skewed XCI patterns are believed to play a role in females with X-linked diseases such as rheumatoid arthritis. To further explore this phenomenon, researchers set out to evaluate XCI skew in different tissues and immune cells using 856 female twins aged 38 to 85 from the Twins UK cohort. The degree of XCI skewing was quantified based on XIST allele-specific expression from paired RNA sequencing and DNA sequencing data of 4 tissue types and 5 immune cell types from identical twins. XIST was used because it is a non-coding RNA expressed on the inactive X chromosome that is heavily involved in the X inactivation process. From there, haplotype SNPs were phased using the program SHAPEIT along with the genetic map B37 and the 1000 Genomes Project. These phased X-linked SNPs were then used for further analysis. The allelic expression of the heterozygous SNPs within XIST were collected and quantified from the paired RNA sequencing and X-linked genotype data using an ASE read counter. From there, ASE values of heterozygous SNPs within XIST were averaged, and the researchers calculated the degree of skewing to understand XCI skewing in different tissue types and involved with rheumatoid arthritis. From the tissue samples of 8 sets of monozygotic twins with one co-twin affected by rheumatoid arthritis and the other unaffected, researchers noticed that the tissue containing the lymphoblastoid cell line, or cells involved with adaptive immunity, have significant differences in skewing between the two twins. These findings supported the researchers' initial hypothesis that X-linked diseases is linked to XCI skewing.
In conclusion, reading Neanderthal Man piqued our interest of the genetic studies in history that aim to answer the question of why genomes differ from human to human, or in the case of the book, human to Neanderthal. Mengelé's twin experiments, although deeply unethical, made a huge impact on future twin genetics research. Twin studies are very useful for genetic disease research and other gene-related occurrences. One study of interest involves X inactivation and skewing using female twins in their tissues. Results from the studies provided information for chromosomal linked diseases such as rheumatoid arthritis and the effects of XCI skewing in different cell types. That concludes our presentation on twin genetics, Josef Mengelé's experiments.